So, welcome to Southampton. My name is Rob Ewing, and what we're going to do is kind of like what we call taster lectures. The idea is that I'll give a lecture, and we're kind of simulating a real lecture that you might experience when you're here. So, in my lectures, I like to encourage people to be interactive. So, if you have any questions or comments, feel free to interrupt at any point. What I'm going to do is I'm going to talk about some of the work and lecture material that we would normally be looking at in years 2 and 3. And the first part of the presentation will be the lecture part, so it'll be kind of the theory of what we do. And then the second part will be more focused on the research, and that research is research that you might be involved in your final year projects. Okay, so just a little bit about me before we get started. So my name is Rob Ewing, and my area of research is in bioinformatics. And bioinformatics is really the marriage of biological science and computer science. So we do things like we analyze lots of data, like human genome data. We use techniques that can measure lots of things in the cell all at once. And we like to analyze those large-scale data sets, all with the objective of uncovering molecules that might be involved in human disease. And I've been doing this for about 25 years, and I've worked in industry as well as academia, and I've worked in lots of different places, different countries, applying these techniques to making discoveries that are relevant to human disease. And today's lecture, what I'm going to tell you about is one of my really favorite research themes, which is about how proteins interact with one another. Now, I put this kind of cartoon down here just to remind you that we're not talking about this, not talking about protein shakes or anything like that you might think of as protein. What we're actually talking about are proteins. And proteins are these fascinating three-dimensional molecules that essentially do everything in the cell. So they conduct all of the biological processes that we rely on to live. So the first part of the talk, I'll tell you about some of the theory about how we discover these protein-protein interactions, so how proteins come together and they interact, and the networks they form. And then in the second part, I'll be telling you about a new research project that we're working on, which is applying these techniques to study brain tumors and to develop a new therapy against brain tumors. So this is one of my favorite slides. I use this all the time in lectures because I think it encapsulates why we would want to study proteins. So I guess many of you will have heard about genomics and the genome. So the genome is all of the DNA in an organism. And if you consider an organism like this that has different stages in its life cycle, so a caterpillar turns into a butterfly, nothing changes. The genome doesn't change between these different stages in the life cycle. So what does change is how the genome is used. And in fact, a large part of that is really to do with the different kinds of proteins that are expressed in these different organisms. So when a caterpillar becomes a butterfly, you get lots and lots of new proteins. This for all intents and purposes, this is a different organism that has a whole different set of proteins expressed. And it's those proteins that allow it to become this new form, if you like. So very different sets of proteins are expressed whilst the genome stays the same. So that's really our rationale for studying proteins, because they're absolutely integral for making us the way we are, for regulating all of the processes and development and so forth of the organism. And I'm guessing that many of you will be familiar with this, which is really where do proteins come from? We know that proteins are encoded in the genome, so genes make proteins essentially. They carry the instructions for making proteins. And you'll have studied, no doubt, how genes are transcribed and how they produce messenger RNAs and how those messenger RNAs are translated into proteins. That's an absolutely critical process in pretty much all life forms. What's not always appreciated is that the story doesn't end down here with the production of protein, right? So once you've got your protein, what happens next? And that's the question that we're really interested in. It turns out that proteins interact with one another. And what I mean when I say that is that they physically come together to produce molecular machines and protein complexes that carry out all of the functions in cells and tissues. And there are many, many different proteins in a typical human cell. If we were to take a skin cell, for example, we could measure on the order of 10 to 20,000 different types of protein in that cell. All of these proteins have got different jobs, and they're all interacting with one another in different ways. And what I'll be telling you about as we move forward is actually how we measure all those protein interactions and why they're really important. This is a nice pictorial representation just to illustrate the fact that proteins come together in lots of different ways. Okay, lots of different molecular machines in the cell. So you might be familiar with the ribosome or the proteasome, where proteins are broken down, or how proteins interact with DNA to regulate transcription. These are all processes that are controlled by sets of proteins, sets of interacting proteins. And so understanding all of these proteins and how they interact is really important if we want to understand anything about human biology and human disease. Now, this is where the network part comes in. So you've all heard of networks, hopefully. So networks really are sets of interacting entities, if you like, and I'll talk about this a bit more in a moment. But essentially, rather than thinking about molecular machines, we can think about protein networks. And these network diagrams, essentially each of these blobs here, these different coloured blobs, represent a different protein. And the links between those blobs are the interactions. So this is really telling us, for example, that one protein interacts with another. And what we do is we try to represent our data in this way as these network models to try and understand which proteins are interacting with which other proteins. Just as a little kind of digression here, we're all used to actually using kind of network information, and network diagrams are really useful ways of summarizing these complex patterns of interrelationships. So, for example, every time we use a transport network, or transfer a map like this, we're essentially looking at a network. And in this case, of course, the objects in the network would be the train stations, and the links between them would be train lines. Okay, we can make network diagrams for lots of types of information. So here's one that I grabbed off the internet yesterday, which is a network of film star collaborations. So you can look up, for example, who your favorite film star actually collaborates with. How many times did they appear together in the same film? These are examples of network diagrams. There are diagrams, for example, of the internet itself. And of course, social media is perhaps the best example of a network diagram. The reason I'm talking about this is that really to illustrate the point that illustrating these complex types of information as networks can be very, very informative. So I just want to give you an example for a moment. So over here, this is actually my Facebook network. Don't laugh, it's a little bit sparse. Perhaps not many friends. I don't use it very often, that's my excuse. But the important thing is here that actually, if you look at a network like this, you can see what we call cliques in the network. Okay? So cliques in the network are actually kind of important structures. If you look at your social media network, if I look at mine, this is my family here, right? They form a clique. And what we mean by a clique is that all people are connected to all of the others, right? Now, if I'm a Facebook engineer and I want to glean information from this, I can take this and say, well, which are the really important key nodes or key people in this network? Well, this one here is very important. This one happens to be my partner. So without even knowing any kind of information, I can look at the structure of the network and I can make predictions about what the role of these different people is in this network. Okay? The reason I'm telling you this is that those very same questions are really important when we study proteins. When we study proteins, we want to know questions like which proteins interact with one another, Are there cliques in the network? The cliques in protein networks often correspond to those molecular machines that I told you about earlier. Okay, so we want to understand the structure of the whole protein network in human cells, just as Facebook wants to understand the structure of all of the social interactions in the world. Okay, so that's obviously a big task. There are tens of thousands of different proteins, but collectively we are making a lot of progress on understanding how proteins interact and also understanding, if you like, the structure of these networks in human cells. So that's kind of the theory. I want to tell you now about how we actually identify these interactions between proteins. And this is an experiment that we do quite standardly in my laboratory. Let's imagine we have a dish of human cells. We grow human cells all the time. Here they are, growing nicely in a petri dish. What we can do is we can take those cells and break them open. And let's say we've got a question about a protein and its interaction partners. One of the things we can do is make use of a tool called an antibody. So you'll be familiar with antibodies as important in the immune system. It turns out they're also really good experimental tools. And the reason they're good experimental tools is that antibodies are specific binders for individual proteins. So we can have an antibody that binds a single protein out of tens of thousands and uniquely binds that protein. And that's really powerful because it allows us to take, this is actually the antibody here, this grey thing. We can take that antibody and we can fish out our protein of interest. And here's the important point. When we fish out a single protein, we often fish out the proteins that are physically associated with that protein. Okay, so when we fish out an individual protein using antibody, we fish out its interaction partners as well. And that's in essence how we build these interaction networks. We use something called a mass spectrometer to identify all of the proteins, to tell us which ones are in this group of proteins, and that allows us to construct these protein interaction networks. And actually this is a very, very powerful set of technologies or tools, if you like, that allows us to do lots and lots of these experiments and build out these network models that we think are really important. Okay, so that's kind of the theory. And as I said earlier, feel free to interrupt or shout something out, comments or questions at any point. What I'm going to tell you about now is actually an application. So what I've just told you about would be kind of lecture material that we might cover in years 2 or 3. In the second part of this presentation, I'm going to tell you about some of the research you might get involved in, particularly in your final year, about how we apply these techniques. And our particular question is whether we can use this approach of uncovering and discovering these protein networks to investigate brain tumors and also develop a therapy against brain tumors. And I want to take a step back and actually, for a moment, focus on what we think cancer actually is. Okay, so cancer is obviously a really important set of human diseases. In many cases, for example, solid tumors, there's a production of tissue in an organ where essentially it shouldn't grow. But our question is really, what drives that? What causes that? And the way we think about tumors and cancer essentially is that it's a kind of a communication failure. And this goes back to the idea of a network of interacting proteins. Essentially what happens in many tumors is that there is an alteration of those protein networks. So you might have a protein that should normally interact with another protein, but chooses instead to interact with a different protein. And that can set off a whole chain of events which can lead to formation of a tumor. So an example might be a protein which normally controls the development of the rate of growth of a cell, happens not to do that anymore, and the cells can grow out of control and produce a tumor. So this is a little bit like if you've ever been, for example, on the London Underground or any other transportation network, and there's a problem at one of the stations, then you know that that problem kind of propagates itself throughout the whole network in unpredictable ways. In some senses, that's really what's happening in a cancer cell. You've got specific changes which alter a protein, and those changes change many other processes as well, because these proteins act in networks. Okay, so this is our perspective, if you like, on how we study cancers and tumors. And the specific thing I want to tell you about is some of the work we've been doing in the area of brain tumors. This is a new project that we've just started. And our question really is, what protein networks are active in what we call brain tumor stem cells? And I'll explain that in a moment. And then our second question is really, can we think of a new way of killing these cells? Okay, so there's lots of reasons we want to do that. Brain tumors are really a really nasty form of cancer, actually often affecting younger people. The median survival for aggressive brain tumors can be really quite short. The current treatment options are not that great. Okay, surgery, radiation, or chemotherapy. So we need new therapies. And importantly for our project, we know that brain tumors are often developing these specific kind of what we call stem cells. So you might have heard of stem cells in other contexts. In essence, stem cells, what that means is it's a type of cell that can continually regenerate itself. And in the context of brain tumors, these stem cells are also very difficult to kill. Okay, they can be hit by radiation or chemotherapy. It doesn't really touch them. So we need new therapies to try to address this problem of these stem cells that produce the tumors in brain tumors. And these stem cells are resistant to current therapies. So let me tell you a little bit how we do experiments with these particular tumors. Now, clearly, we're not doing experiments on people. We are using tissue from human patients, and we're taking that tissue and we're producing what we call cell lines. So this is really taking a tiny amount of tissue and being able to extract cells from that tissue and then grow those cells in culture. And that, if you like, is our model system. That's what we work on. All of the experiments I'll talk about are done in these cell lines. And importantly, when we grow the cell lines, we can, if you like, mimic the way they might grow in a tumor, right? So they produce what we call tumor spheres, which are sort of masses of these stem cells that behave a little bit like tumors actually in the brain. So they're good experimental organisms, if you like. How can we kill these cells? I'm going to tell you very briefly about an idea we've got that comes from our collaborator, that we can actually use a virus to kill these cells. So, has everybody heard of Zika? Yep, so the Zika epidemic, the most recent one at least, was in Brazil in the years 2015 to 2016. And actually there was a unique thing noted about the Zika virus in Brazil a couple of years ago, and that is that if it infects pregnant women, there was a high likelihood that their babies would be born with a condition called microcephaly. And microcephaly essentially is a smaller head and brain. It's a really nasty birth defect. And this really was noted in a small town in Brazil several years ago, and has really led to a lot of research into the Zika virus. So the Zika virus is primarily spread by mosquitoes, and it really occurs across the world, but Brazil has been really an epicenter for the Zika epidemic. We know that Zika works. In fact, we know that Zika works to cause these birth defects by infecting what we call the neural stem cells. Those are the normal brain cells that allow the brain to develop. But here's the thing. We also know that Zika will very specifically infect brain tumor stem cells. Okay, so the question we began to ask ourselves is whether actually you could use this virus to try and target these specific brain tumor cells that we find in these aggressive tumors. And I'm going to show you a little bit of work that suggests that is possible. So these are going back to our cultured cells. Remember, that's our model here. And this is just to show you the effects of Zika. So this is a representation of the Zika virus. And what we've got here are different types of cells. At the bottom here, we've got normal brain stem cells. Okay, and actually, the Zika virus has very little effect on the growth of those tumor spheres. But if you look at the very aggressive tumor stem cells, you can see that the Zika virus essentially kills those cells very specifically. So we know that Zika can target these brain tumor cells, and we know that Zika can kill these cells. So the question that we're really addressing with the techniques that I told you earlier about is, how does this happen? How does the Zika virus kill brain tumor stem cells? And this is the kind of experiments that we're doing. So what we do is we take those brain tumor stem cells in a dish, we infect them with the Zika virus, and then we use antibodies, just as I explained earlier, to target the Zika proteins. And then we pull out the Zika protein from the cell, and we look and see which human proteins are attached to it. And what that tells us is that it effectively tells us about the protein networks. It tells us about the Zika protein, what it's doing in the cell, and also which human proteins, which host proteins, it's interacting with. So we can do lots of experiments like this, and this is allowing us to understand the protein networks that are affected by the Zika viral proteins. So this is really the essence of our technique. We want to understand how the Zika virus infects the human cells and how it changes the protein networks. Okay? So this is ongoing work that we started last year. I want to finish by just giving you a slightly different perspective about science. There's really two key things that science needs, in my opinion. It needs lots of things. Firstly is people, and this particular project is a collaboration with a friend of mine in Sao Paulo, Brazil, who has done a lot of this Zika work. So we're working together to try and resolve this issue and try and develop Zika as a therapeutic. And the second thing it really needs is the right equipment. And actually here at Southampton, we're really very lucky, because 2 floors up from here, we have a centre for mass spectrometry. So the mass spectrometers are those instruments that I told you about earlier that allow us to identify the proteins in the cells. And we have the best facility in the south of England, one of the best in the UK. So we're really, really well resourced with respect to instrumentation. So putting these two things together with a little bit of money, we can actually potentially do some good science. And hopefully we can involve you in that science as project students if you come to Southampton. So any questions? Yeah. So basically what we do lecture-wise, we kind of teach the theory, or at least I teach the theory of mass spectrometry, and then with actually the several projects that we've run involve using the mass spectrometry, getting the data and analysing it. So we do a lot of data analysis, a lot of that kind of stuff that is associated with mass spectrometry.